Hello, my name is Christopher Sontag, and today I will be presenting my undergraduate research project, Differential Expression Analysis of Duplicate Genes in Primate Brains. Dr. Chain is the principal investigator in this project, and it's also important to mention that the data for this project was taken from Kremiva et al. 2020, and some of the methods used in the analysis were adapted from Cote et al. 2020. The most common source of new genes is gene duplication. When this occurs, two copies of the same gene are initially created, usually due to an error during DNA replication. Duplicated genes usually result in the loss of one of the two resulting genes. However, sometimes through DNA mutation, one of the two resulting genes, also known as paralogs, differentiates from the original gene and becomes a new gene entirely. This creation of new genes can be measured based on how much they are active or expressed between different cells and tissues. We've been able to investigate the differences of gene expression between tissues for years. However, looking at differences between cells is a very recent development. Our research question was, are the changes in expression of duplicate genes the same or different across species? To answer this, we use single-cell RNA sequencing data, which allows us to determine differences in expression between cells rather than traditional RNA-seq, which can only reveal differences of expression on the tissue level and may leave out key findings. The data we used for this project was taken from Kremiva et al. 2020 and consisted of single-cell RNA-seq data from humans as well as 3 closely related primates, bonobos, chimps, and macaques. To answer our question, we first need to categorize duplicate genes based on the ways their paralogs are expressed. We used many classifications to categorize the duplicate genes. However, we are most interested in class 4 duplicate genes. Duplicates were categorized as class 4 when reciprocal expression was seen, where in one cell there was a bias in expression towards paralog A, yet in another cell there was bias expression towards paralog B. Class 4 duplicate genes were often species-specific, meaning that if a pair was classified as class 4 in one species, it is probably not classified the same way in any of the other species. In regards to the figure, the pink boxes represent the number of class 4 genes that are only present in one species and shared between 2, 3, and all 4 species. In general, the number of pairs in the same class decreases as evolutionary distance increases. This means that pairs are more likely to be in the same class when comparing two closely related species versus two species that are not closely related. In regards to this figure, it is formatted so that the further right on the x-axis, the more evolutionary distance between the species. The y-axis represents the number of pairs in the same class. Thank you for listening.